Hi there, welcome to another edition of TokStocks. I'm your host, Keir Reynolds, and today we're lucky enough to have Guido Baechler, Executive Chairman with Telo Genomics Corp. Trades on the TSXV Venture Exchange with the ticker TELO, Telo. Hey, how you doing today, Guido?
Hey, great to have you. Great to have you to be on your show. It's great to really see you here.
Hey, excellent. Well, thanks very much for joining us. I really look forward to sort of unpacking things. Before we get into Telo and what Telo's all about, maybe you could, for those of my viewers that have never met you before, maybe you could explain a little bit about who you are, what's your background?
Yeah, absolutely. So I've been an industry veteran for over 30 years. I spent always in the diagnostic world. So I spent close to 20 years with Roche Diagnostics, one of the world's leading medical device provider in Europe as well as in the United States. And I had a variety of roles within Roche, really, from development to commercialization to program management. So I've seen really a lot of programs, from early research stage to very, very complex, PMA and FDA submissions. So I got a really good education there before I then switched and started joining and then ultimately running startups. So I'm currently running a NASDAQ traded molecular diagnostic company. But as you introduced me before, I'm also the executive chairman of Telo Genomics, and I'm very thrilled about what Telo is doing. Actually, really, really thrilled to share more information with you and your viewer about the potential of the powerful technology that Telo Genomics actually is developing.
Where are you based today? Where do you live? Where's home for you?
So besides planes, I'm based in California, Berkeley, California, where I moved over to California in 95. Thinking we'd go over there for a couple of years, and that's now over 30 years. So I do spend, Telo Genomics is based in Toronto. So I spend time in Toronto with the team. We have a wonderful facility in the Marsh Center in down in Toronto, where we actually provide our services to clinicians in US as well. But we'll come to that in a minute.
And how long have you been with the company?
I've known Telo for several years. It's been pre-COVID, where I first met the team in Toronto. And I also met Dr. Sabine May, who is the founder and interim CEO of the company. And Sabine has, over 20 years of experience. She raised a lot of non-dilutive financing for the company and really developed this technology extremely robust. She also has applied this technology across many, many disease areas, and I'll talk about that. So it's a platform technology that Telo Genomics has developed, and I can't wait to give you more color on that.
Well, excellent. Well, thanks very much for giving us a bit of a background on yourself. I know you're just itching to tell us a bit about Telo. Could you give, so for many of my viewers, this will be the first time they've ever heard of Telo Genomics. What is the company? Can you give us a bit of an overview?
Absolutely. So, basically, maybe I want to go a step back a little bit and give you a little bit more color. So, it's a genomics company that uses microscopes to look at molecular levels of chromosomes. And so many of you know chromosomes, we all have, chromosomes. And so there's actually a very important part, within what's happening in the human being with the chromosomes once they're divide and are separated, and so there's an aspect that's actually happening in the human being, and we call this genomic instability. Genomic instability is something that actually happens when individuals get sick. When individuals are developing cancer, for example, then the chromosomes rearrangements doesn't happen correctly. And it's really important to know, so that genomic instability is a terminology that's used very broadly to really identify if something is going wrong with the individuals. So when you look at the chromosomes, and again, the way the X shape many of them have, the end point of the chromosomes are telomeres. And it's like when you have a shoelace and you have at the end of the shoelace, you have like a plastic around it that protects the end of the shoelace. Very similar, you have now telomeres are actually wrapped around the chromosomes to keep them in shape. And so, and the reason I'm telling you this is it's important because what Telo Genomics is doing, we're actually analyzing the telomeres of the chromosomes at an extreme low levels with microscopes. And then we have built algorithms around it to help predict what actually happens to you as a patient if your telomeres are starting to change. Many of you have heard, the discussion of telomeres when you get older, but the telomeres are getting shorter. So telomeres are being used quite a bit in discussions of age. Then people want to behave differently to reduce the shortness of the telomeres and all that. That's a very simple way of looking at it. And telomere has really gone way above and beyond and developed now this technology that allows me or us to look at individuals and measure their telomeres and actually have predictions. For example, we can help identify if you are a cancer patient, how long a specific cancer drug would work on you. We can also help identify individual cancer cells to see where the therapy should stop, and so on. So Telo Genomics really developed, I would say, an industry-leading platform to really assess genomic instability through the measurement of telomeres that we can look with our platform that we run in our clinical lab. So, it's really, again, I think the key is think about the chromosomes, right, that each of us have. And then the end piece of the chromosomes, think about a shoelace. And it's really the change, telomeres get shorter, you get more telomeres, you get less telomeres. They're starting to stick together. These are all elements that we're looking with our when you go to our website, you see some really cool videos that shows you, and then all is tied into machine learning algorithm at the end, then ultimately creates clinical results that then a physician can actually use to treat you or the patient, with better therapies, not stop therapies, or putting you on beginning of therapy. We didn't know whether we should do this or not before.
Hey, excellent. Thank you for giving us that good overview. Even I've heard a little bit about the discussion on telomere and telomere length and being able to predict things like aging and stuff like that. So very, very cool. I really appreciate you giving us a good education and an overview on that. How is Telo Genomics kind of set up to be able to sort of leverage that kind of technology? And maybe you could tell us, does the company have patents or what's it sort of doing to really develop sort of like IP or a product sort of out of this?
Right. So we have Telo Genomics, again, works very closely with Sabine May, who runs a cancer center in Canada, and we license all the patents from her. So we have a complete panel of license for specific diseases. So it's very important, as you can imagine, as we start developing. So Sabine is really the research center, and we're converting that into Telo, who then develops these products into products that we can then sell and provide to clinicians. How we're doing this? So we actually have a laboratory in Toronto, a CLIA/CAP certified and ISO certified laboratories that allows us now to offer some of those tests to physicians in Canada as well as the United States. That's really important. So, our approach is to be able to, it's really this CLIA/CAP certification that was critical for us to be able to now validate specific tests. And one example is we have already validated a smoldering myeloma test. So what is a smoldering myeloma test? So if you are an individual who's been diagnosed as a potential risk candidate for achieving multiple myeloma, which is a really ugly blood test, a blood cancer. So you may be categorized as a smoldering patient. Smoldering patient, and when you go to your multiple myeloma physician, they're not 100% sure whether they should start therapy or not with you. There's a potential that, if you are a smoldering patient and you get therapy, you may never progress to being a cancer patient, but I'm not sure. And our test actually has shown, with multiple studies that we've done with the Mayo Clinic, we have a very high accuracy of being able to say, Keir, you should be treated, or Guido, you should be treated, not treated. So it's very powerful. And this test is actually offered in our ClearCap certified lab. That test has IP around wrapped it, so we're protected. And we're starting to not sell the test, but we're offering it to clinicians in the US. And why are we doing this? We really want to create awareness with multiple myeloma physicians that there's a test out there that they can start using to make clinical decisions. And so that's the process that's going on. That's one example. The second example is, which is an even bigger opportunity, is called minimal residue disease. So what is that? Minimal residue disease is if you're a cancer patient and you're going on therapy, there's a time when the physician asks, should I still treat Guido, or is Guido actually okay now? Is he cured? And then at what stage can I stop therapy? And that's since in 2024, FDA made some new guidelines that says if for example, if my cell count is below a certain level, then I can stop therapy for me, right? And sometimes I go in remission for a while, but then I relapse. And it's unclear how long I relapse. And what Telo Genomics does here as well, that test is in development, it's not yet validated in a CLIA lab. What we can do is we can not only count my cells, but I can profile my cells. So I can see with our microscope and our software, I can see that Guido still may has few cells, that somebody may say, well, stop therapy. But a lot of cells are actually aggressive. Don't stop the therapy. So it's a really new approach of not just counting like what everybody else is doing, but now also profiling the cell. Like say, hey, do not stop. So that's a simple way of looking at it. Of course, it's more complex, but the idea is that we're providing a new tool for patients in our multiple myeloma. Again, once we have the test validated, we also want to start offering that to clinicians and patients in the US and Canada as well. So that's another application that we wanted to get into our clinical lab. The next one that we're discussing is prostate cancer as well. Many of you, again, I'm a guy myself. It happens, and what just happened with Joe Biden as well, right? He was actually one of those candidates, right? And the Gleason score is basically how you add up whether you are at risk. If you're Gleason 6, you are a low risk. If you Gleason 7, you are intermediate risk. And it's not always clear what to do. And Sabine and her team has shown in the past as well, the clinical trial, that we can do without a biopsy, have a pretty good accuracy to predict whether you should do a biopsy or not, whether you are at higher risk of progressing or not. So again, another application that is in the works. And the last one I want to talk about is Alzheimer's. And that's a brand new one that we haven't talked much about it. But Sabine May has done a lot of research in the past as well, which we're now starting to get into. Telo Genomics as well, and we will have more discussions as we move on. We can wear a very simple buccal swab, have a very good accuracy of actually identifying potential mild Alzheimer's. And I don't need to tell you, okay, how big that is. So that's another opportunity where we can show that with a very simple test, we can be very predictive. Clearly more work needed, more studies be needed, but the potential of that as well is incredible. So, and I think that kind of reflects why I am so excited about Telo Genomics. It just has, I've seen with Roche, I spent a lot of my life with PCR, right? Which before COVID, almost nobody knew what the heck was PCR, and now everybody knows it when they stuck it up your nose, right, and do the testing. And so PCR I was really at the beginning of PCR where
it's all your fault.
It's no, don't, please don't. But it was interesting, right? It was very beginning of automation, and now it's available. And so with Telo Genomics, I see also here potential of this. It's kind of it's a secret jewel here that we have, that we're slowly starting to move forward into the clinic and create more awareness, because the potential is just incredible big.
Well, that's a very interesting little anecdote about yourself being involved fairly early in the PCR, because yes, none of us had ever heard of it before that awful sort of global pandemic. But I guess we were all very lucky that such a test existed, so that we could rapidly determine who was sick before symptoms sort of took hold. Right. So anyways, very interesting stuff for sure. So if we circle back around to Telo here, so Telo is really, sort of like not a traditional sort of biotech company. It's more of like coming out. It would fit into the category of like a diagnostic or testing type company. And so typically, what are some of the value sort of stage gates that you have to get through before you could start selling a test? You need to go through traditional things like clinical trials, or what other sort of process is there to go from, hey, we believe our testing platform could work for this illness or condition or disease. What are the steps that you'd have to take it through before you could actually monetize and sell that test?
Yeah, that's a great question, Keir. So fundamentally, we start off with studies, right? You may on a research level, like some of the work that Sabine has done is on a research level, you start creating the proof that fundamentally the technology works, like I just said on the smoldering, for example, right? So you start testing and see, like what we did with Mayo, you get the first batch of samples where we know the outcome, right? So we know the outcome, and then we run the samples, and then we train the algorithm, right? So basically you almost look at, and you show that actually it fundamentally works, right? But then you need to do in the second study, for example, where then you have blinded samples. You have no idea what the results are. You run your samples, your technology on your samples, and create results. And then you send that result to somebody else who then correlates your results with the results from the clinicians. And that's called the validation. That is the second part of the study you have to do, in our case, to show that the technology actually works. And so there is, it depends on timing, how long the study takes. It can take 6 months, it can take a year to get the different studies done. Then you also have to do a lot of documentation. And in our case, we offer it through our ClearCap lab, right? So that allows us now, with the proper procedures in place and proper validation, to offer that test based on the claims we can make based on the studies. So that is the ClearCap approach, right? Another way you can do it, which we may do in the future, we can go through more like a 510K approval, where we then basically able to validate our technology to sell it to many labs in the US, right? So this is, for example, what Roche does. Roche has hundreds of machines all around the world, validate the test, and then anybody can run it. On the other hand, there are laboratories that test own lab in their own lab, and you can only run it in a specific lab. So there are different ways to do it, but everybody always has to do extensive clinical trials to show that whatever you claim can be backed up by clinical data that you run in your own lab.
And so are there, just to get a little bit more granular on that, a lot of times we hear if there's like a drug treatment platform that a biotech company is looking to develop, they go through several phases of trial before we get to like human clinical trial, and there's a number of phases of that, and depending on the phase and whether you would be deemed successful, can be a big re-rate on a stock. Do we have that same traditional kind of path to success that for sort of a testing diagnostics company, or is it
It's quite different. It's quite different, right? We don't do it because we don't inject anything to humans, right? So like there's no safety, right? So it's not because we are taking blood. We're taking spinal fluid, we're taking elements from you and test it, and then often aid in the diagnosis of a disease, right? So from that point of view, the process is simpler, right? So, we don't need to go through stage 1, 2, 3, all these phases. It's pretty much, you have to do that. So there's a clear process what you have to, for example, you have to test your materials. You got to make sure you have reproducibility, right? So if I sent my sample in today and I sent my samples in a week, there should be similar results, right? So all that work has to happen behind the scene before you can actually offer a test like this. But it's not, so when you look at a pipeline, right? So we can certainly say this pipeline is in research now, and then the pipeline is now in validation, which means then we kind of frozen certain aspects of the test and the algorithm, and now we're going to validate it. And once it's validated, then we can actually start the commercialization with clinicians. And at the same time, then we have to have discussions on reimbursement, right? So which of course happens hand in hand, because you want to make sure, you have, again, once you start commercializing, you must have the whole discussion of reimbursement already established as well, because that's a separate process, how you want to make sure your test actually gets reimbursed, at the appropriate level based on what kind of service you're providing.
Excellent. So I guess to kind of summarize and think of this from like an investor point of view, you guys have a simpler process because there's no safety. It's also less costly and time and
that's
like
right.
That's the problem with some biotech companies, especially if it's a startup. You need millions and millions and millions and millions of funding because you have to go through these sort of laborious, expensive, multiple sort of human clinical trials, which when you're trying to inject something into somebody or have somebody ingest something, there's a lot of safety that have to sort of go into that. So as soon as you're drawing blood and really testing it, the path is really about if you're able to show repeatability and if there is actually a result sort of there,
right?
And so there's now there's an established process for that type of diagnostic sort of company. You mentioned that there's maybe two pathways to do in order to, Roche has sort of a platform that they generally follow and other companies would follow. And then there's another process which you guys are kind of currently following.
Exactly. For example, I'll give you another example, which is in our business, well, is Exact Sciences, right? Exact Sciences does a lot of colorectal cancer testing. They have a big CLIA lab. They have a big central lab, and everybody ships everything to their lab to do testing there. So that's the CLIA centralized lab approach, right? Where Roche has instruments, any lab can do it, right? So it's like what we did with COVID. Think about it. If COVID would have had one lab, it would have been a nightmare. The technology was spread all over the place and everybody was doing it. So what Telo Genomics does initially right now is a central lab, right? So if somebody wants to test, have to send it to us. And that's the most effective way to get a product into the market. It's generally faster than the other part because it's easier to do a test in your own lab rather than making it work in 100 labs. Think about it from that point of view.
Right. Gotcha. That makes it clear as mud. Thank you for explaining that in such great detail. So you mentioned a bit about kind of some of the applications. I guess you started with this smoldering myeloma. Was that kind of your first kind of platform and would that be the most advanced in terms of getting it close to a place where there'd be commercial? The ability to commercialize that?
Yeah, so it's the one that's, we have Hodgkin's lymphoma as well validated, but let's focus on smoldering. Smoldering fully validated in CLIA lab, and we're running it right now with institutions in the US where they're basically sending us samples. And this is not commercial yet. It's really a pre-commercial activity to create awareness of our technology. Right, so that's a very important part. But the next step will be is the MRD, which is actually, I think, if you look at what Adaptive, for the colleagues who look at the Adaptive company who's looking at also running MRD in multiple myeloma, and they have massive revenues over 100 million on that specific product. And we believe we have a better product, because we're not just counting, but we can also profile the samples. So there's a big potential. That's why we're pushing this really hard now to get this test validated in our own lab. Again, that's something that as we talk about in the future, I'll give you more updates. The team is really working very intensely on that, trying to get that, and that should happen within the next probably 2 to 3 months that test is also validated. We will then release it as well. And we'll start then also, similar to what we've done with smoldering, start having it being used in the US. We're doing that already today as part of, we actually have 2 clinical trials ongoing, which we already announced. We also do a kind of comparing our technology to Adaptive, which is kind of the gold standard today because FDA approved. So it's fascinating to see where this is going. So again, I would like the viewer really to focus on MRD. Because that is a big opportunity. And then third one is also newly diagnosed, where we basically can predict when you have relapse with certain therapies. So those are really, it's a whole portfolio of multiple myeloma products, and clearly we have, it's not all validated yet, but once it's done, we certainly have the most comprehensive, tool for helping manage multiple myeloma patients. And that's something we certainly will then also start having discussions with reimbursement experts, etc. So this is also ongoing this year. Besides the development and validation, we are planning to also start the initial discussions with payers about, what would such a test be reimbursed. We don't have those answers yet.
Well, thanks for walking us through that. Well, so at this point, the company's not generating any sort of commercial revenue from any tests. At what point do you think that would sort of change and there'd be, revenue potential for a company like this?
I think that our goal really is to, make the platform, make companies aware of it, right? So again, for us, at the moment you go commercial, you have to hire a lot of sales reps, which is not inexpensive. And so our goal is to try to network with these big centers in the US, because once they work with you on this kind of earlier study approach, and then you can raise the sales flag, and without having a long sales rep, you already have a broad, at least a base of clinicians who want to use it. That's kind of the approach, right? Because we don't want to go out and hire 20 sales reps. It's very expensive. Our approach really is to network with this institution, get them live with our product in a research setting, and then flip it over and start having a commercial test. But that's certainly not going to happen this year.
Gotcha. And so, one of the interesting things with cancer is you typically have, specialized clinics that would be kind of the world authority or and a lot of that stuff is kind of happening in the US. Do you have any sort of like key partnerships that would be considered leaders in terms of research and bringing new sort of like treatment to some of these disease areas that you guys are focused on?
Yeah, absolutely. And again, some of them we will announce shortly. But one of them that we have announced and lots of work done with is Mayo Clinic. And here as well, we've working with Kumar, who is, really leading a lot of the activities, and also with Ken Anderson from Harvard. These are the guys who really are the key drivers in what's happening in myeloma, plus some other ones as well. These are not the only one, but those are the two we work very, very closely with. And they're guiding us on how to approach those diseases, like the MRD, the minimal residual disease, for example, is one of them that came through some suggestions from those guys. They saw that what's out there today is just not good enough, and they could tell that our technology could potentially fill a void, and we are pushing forward with that now. So we clearly see, where there are clinical gaps, and we are going to push hard to provide products based on feedback from clinicians using it every day.
Well, thank you for giving us an overview on that, and congrats on getting some elite partnerships there. Look forward to, you mentioned there's a few others to sort of get finished and announced, so we look forward to reading about those when they're done. Now, why don't we turn our attention a little bit to kind of the competitive landscape. So you guys have a test, you guys have a process for testing that's sort of been developed over a long period of time through Dr. May's kind of research, her being sort of the founder of kind of the technology and the platform, the company. What sort of traditional what are you competing against? Are you competing against sort of like biopsies? Are you competing? What other sorts of things are you kind of competing against? You did mention a little earlier, there's a company Adaptive that have something. Right. So maybe you could, for us layman people, if you could drop it down to our level. What are you guys competing against, and why do you think your platform's better?
So again, on the telomere technology itself. So let's break it into two sections. One, so there are other technologies that can measure telomere length, for example, and one of them is PCR, right? So there are others as well, but those are technologies that can work on some elements of telomeres, but they cannot do individual cells. They measure a group of telomeres and not individual ones. So that's where we see a huge advantage. And they can only do length. And as Dr. May has shown, in many times, length is not important. Number of telomeres, for example, are we've seen, for example, in some studies, more important for Alzheimer's, or how many, again, where the telomeres are within the space, whether they're clogged together, and how bright they are. So there's no technology out there today that I'm aware of that has the breadth of providing different type of telomeric biomarkers, if you want to call it like this, that can be applied. Again, Sabine has shown this across. Now on applications, right? An MRD, for example, want to come back to that. There are flow cells, companies that count cells. There are sequencing technologies out there. So there's a variety of technologies out there that are being used, and some of them are, for example, not as sensitive. So there's a measurement how many, for example, look in a water glass and how many molecules you need to have in there. If you fish it out to detect it, some are more sensitive than other ones, right? Some are clearly not very sensitive, not as sensitive as we are. We will show the data. So that's one element, right? So there's the sequencing and other technologies. Sometimes they're not sensitive, but the most important part there is, as far as we know and understand all these technologies, no one of them can actually truly measure the biology of individual cells, which is so important, right? So that you can identify patients that are actually have aggressive cells and on. I think that's the key in there. Again, you can look it up. There's other, again, on the telomere technology, there are other ones out there, but none of them is broad. And length is good, but length is not everything. You need to have other elements in there. And on the applications, again, here as well, there's multiple sequencing and other flow cell technologies out there, but they're just not as good they may get as sensitive as we are, but they're not as good as identifying and profiling individual cells.
Well, thank you very much for giving us sort of a breakdown on that. So you mentioned, just to kind of reiterate, and then we'll kind of move on from here. Maybe you could outline again, just for my viewers, what platforms are you currently sort of actively advancing? So we got smoldering multiple myeloma, SMM,
right,
and then you've got your MRD, your minimal residual disease, if I'm not mistaken.
That's correct.
You got prostate cancer and you got Alzheimer's. Are there any other potential applications that are sort of on your pipeline or that you're looking at? And which of those that I just talked about are sort of most active, and are all of them active?
So right now, so I, as you and the viewer appreciate that, we don't want to do too many things. We want to do a few things and want to do them right. So right. Multiple myeloma is core for us at this stage. We're testing smoldering samples from US and some Canada as well, because we have it validated. Then there's the MRD, which is an ongoing trial that we're doing, measuring all samples from the US and from Canada. Big focus. So I would say smoldering is validated. We're going into the pre-commercial, right? Building customers in the US. We have MRD in development, probably the biggest focus of the company right now, because we want to show that is truly superior to what's out there today. With potential going to the FDA or getting, a breakthrough device designation. If the data is good, we want to do this because we want to get the product out there. And the third is newly diagnosed, right? So being able to predict when you're going to relapse. So multiple myeloma is key. Next one is Alzheimer's. We're starting that as well with the team to get that fully validated. Again, MRD will be priority number one for new products, and the next one is Alzheimer's. Show that, and we will start publishing more work on that as well, as Sabine will do that as well. And then prostate is the third one, where we're exploring right now to do some additional studies, potentially with strategic partners that have samples available, so we don't have to do another retrospective study. So those are the three ones I would say you should focus on, and we will focus on.
Are there any other?
There are 20 other applications out there that one could do, but quite frankly, with the resources we have, we want to focus on those and get those, further developed, because that allows us to, again, it's about when you have a platform like we have, you can do too many things and end up doing nothing right. And so our goal is really to stick to the few ones and get them done right. And there's plenty. We have Sabine has many other applications developed in a research program that we ultimately absolutely we're thinking of getting into the market, right? So we have lung cancer, for example. We have some stuff for breast cancer, leukemia, brain tumors, right? Thyroid cancer, you name it, cervical cancer. So Sabine has done work across a really broad spectrum. We just can't do it all. We want to focus on a few of them, select ones, right? Get them validated, maybe do some partnerships with strategic partners and focus on the next one. It's really this is a pipeline development that we're doing, but those are the three areas we're focusing on right now.
Well, thank you for that. So at what point would, a larger biotech company, find this company interesting as a potential takeover target? Where do you think you have to get to? And would that be sort of like the logical sort of outcome, or do you believe that you're going to be operating this business and generating revenue through basically processing tests, selling your test?
Yeah, so I don't have the crystal ball, but from my past experience, right? So basically, when I look at it, you want to have some meaningful applications that could be very interesting to the company. What I did in the past, right? So you have a platform, you can license one disease area, for example, for one partner, and then you can work on something else, right? So of course, somebody may want to take it all out, but I think that the more realistic approach is that somebody is really interested, because our technology can be very easily converted into another lab, for example, right? So if there's a big lab in the US who wants to take on the multiple myeloma, product, right? Because we have a whole palette of product in multiple myeloma, they may take that on, take the whole technology on, but just for that specific disease area. So in our case, you could do a potential licensing deal with a strategic partner to take on that area. We could do something else, right? So, but ultimately, it's hard to say whether somebody will, because it's so broad, and it's an opportunity for us too, because there's certain things we absolutely want to do, maybe want to do ourselves. So you get some additional capital in the organization through licensing deals on one disease area, and you focus on something else. So, but ultimately, I know what the investors want to do. So we hope that ultimately will then trigger to a full acquisition of the company, but we'll have to see where this goes.
So what would make somebody like yourself, jump in and become executive chair of a company like this? You worked with, obviously some very large, like Roche, obviously massive company. What makes you look at a little small, teeny, MicroCap Canadian listed, company? What got you so excited?
I think it's the speed of things, how you can get stuff done, right? So I've been long enough in big organizations and I know what is required. But there are different ways of doing it, right? So large organizations have lots, groups, debates, and quite frankly, the big organizations generally don't innovate. They watch and buy something else, right? So, and I've gone through the process of the big organizations. I love the small, younger teams, right? The things I've learned, all the battle scars I have from my past, and really leveraging that into a smaller organization. Do not make the same mistakes again, and really stay clearly focused of something that ultimately has shareholder value. And I think here that's where I can really, and I love to do that. And I think that's where I'm good at, really working with, leveraging my experience with big companies and my smaller one I run, and then really work with the Canadian team to really get that really powerful technology, broadly used and address all the important unmet clinical needs that we can address with them.
What does success look like specifically for Telo, the company, over the next 6 to 12 months? Generally, what are the sorts of things that we as investors should be paying attention to?
Right. So I think that the important part is what I would look at really is the further validation of these assets we were talking about before, right? So really looking for the MRD, clearly for us that is a milestone, being able to validate the MRD with our technology, that we can prove to the industry that, hey, there's a new technology out there who can do minimal residual disease equal or better than what's out there today, right? So I think that is a big milestone for us. And also, there's a lot of development going on in the Alzheimer's space, right? So where we clearly, all of us will probably, hopefully not too soon, start forgetting things, and being able to early diagnose Alzheimer's. Now that certain therapies are around there, specifically on the reduction over the amyloid growth, is very powerful. So again, here, developing tools that are simple and inexpensive used in a broader community, that's another one. So again, look out for that as well, that you hear a lot more. So the next 6 to 12 months clearly is get MRD validated, get the data out the door, get the data shown, and also work on Alzheimer's, but then some other strategic partnerships that we're going to go in, because we have some really, really powerful new clinical partners that we're starting to work with, and those will be announced as well in due time.
What's the sort of size of market of some of these things? Like, for example, for multiple myeloma, how many tests could you sell a year? What's the size? What's the TAM? What's the total addressable market size?
Yeah, so again, just be very specific on what Adaptive does, right? So they have over 100 million on just MRD. So these markets are massive, right? So again, they're not, especially on this is for the heme cancer, right? Myeloma is the biggest one. So again, this is the reason why we selected it, because it is actually a massive market. And if you are a smoldering patient, you should do repeated testing, right? Because you shouldn't do this once. You do this once and then you will keep doing it after a certain time. The frequency to be determined. I think we assumed about twice a year, which creates a very, very vast market. So I don't like to just put numbers out there. I just want to see what's real today, and that's Adaptive. And if you just get part of what they're doing and go in that direction, this is easy revenue, hundreds of millions of dollars.
Excellent. You mentioned right at the hop that your technology using microscopes are able to see at such a high sort of resolution down to the individual telomeres, and then you're using some analytics and things like that. How much has AI sort of involved in your sort of company?
So not AI at this stage. We have some algorithms that helps us so looking at the different parameters and selecting the right one. But we're starting to evaluate more cell selection. It's kind of very difficult to select all the cells, and there's some really cool tools out there that we're starting to evaluate. So I think the algorithm we're using is more of a machine learning approach. And not real it's always the definition, what is AI versus machine learning, but we're certainly starting to look at more automation of our process and using AI to help us select some of the cells, and that's certainly something we are going to explore as well in the coming quarters and years to come.
In terms of your testing, is there any unique sort of like hardware or technology that's sort of needed in order to do it, or is it more just the process of what you're looking at?
It is more the process, right? So because, a laboratory who has microscopes and runs the FISH process, they have microscopes, and most of them are good enough for us to use. And then the software is just the software. So it does not need a massive investment. That's why we like the approach. It can be relatively easily converted to another lab without massive investment. And it makes it simpler. Because there's, again, you don't have to install equipment, and there's no capital investments that companies and laboratories have to do.
Excellent. Well, that's good. I'm glad we got that out there. That's a whole other business on its own, and not one that always plays out very well with having a. So great to hear that. What are some final thoughts you'd like to leave viewers with here?
Well, first of all, I wanted to thank all of you for listening to my excitement about Telo Genomics. It is a hidden jewel. I know we were some struggling. When people look at the share price, we're not happy about it. But at the end of the day, we have a good team together. We have great new applications, and we're keep watching us. I think you're going to be happy where we're going. Because we have some really exciting news coming out, and some of them I shared with you today. But this is what we're talking, what we're planning to do. And again, please please keep watching us, and I'm happy to come back here to give you some updates in the next 4 or 5 months, see where we are compared to what we were today. But I think we're onto something, and so I really appreciate everybody's support to help us get that technology really validated and into the clinic to help patients being treated better and more effectively.
Well, excellent. That's a great way to sort of end off. Guido, it's been really great to meet you. You really obviously come from a great sort of deep background working with big companies. So I would say probably understand better than most kind of what they would look for before looking to partner with kind of a startup, and then how to guide that startup. So anyways, they're very lucky to have you, and great to meet you. And thanks very much for spending some time and answering a few questions.
Same here. It was great to meet you, Keir, as well, and looking forward to coming back.